Hi, my name is Safia Safa-Tahar-Henni. I'm a PhD student at IRIC from University of Montreal. The subject of this presentation is the identification of potential therapeutic targets in AML through the integration of genomics data and small molecule screen results. Acute myeloid leukemia is a cancer that starts in blood stem cells. Research on AML has identified recurrent mutations, such as the translocation of the KMT2A genes, which are generally associated with a poor clinical prognosis. When this gene is mutated, it fuses with a protein partner. Over 120 different partners genes have been identified. The different fusions of the KMT2A genes are associated with variable survival rates. Recently, we have developed a new single-donor leukemia model that enabled the development of the human AML from normal cord blood cells. The presence of the KMT2A fusions is sufficient to develop AML in our models, but this is not the case in patients which present recurrent secondary mutation. The hypothesis we made is that if we compare the effect of therapeutic compounds between models and patients, we will be able to highlight the impact of recurrent secondary mutations despite the heterogeneity of the patient. In order to better understand the genetics behind this leukemia subgroup, we will perform a high-throughput chemical screen of 11,000 compounds at a single dose. This will be tested on 50 samples from patients, models, and cell lines with different rearrangement of the KMT2A gene. On this sample, we will also have access to information on expression and exome. We will be interested in compounds that will have an impact on the cells of patients, but not the control. This compound could be used as a treatment, as well as a compound that will have a different impact for the different KMT2A gene fusions, and compound that will have an impact on patients, but not on the models, and vice versa, in order to understand the impact of secondary mutation present in patients. The compound identified will be validated by a secondary dose response screen and then investigated using functional tests. The limiting factor of this project will be the number of compounds that we can investigate. To highlight compounds that have a significant impact on cell growth, or HITs, the output value of the high-throughput chemical screen must be processed in 2 steps. First, the raw data is normalized, then we need to select the HIT. So the 11,000 compounds will be added to the cells in 36 plates of 384 wells for screening. Each plate contains 4 columns of negative controls on the edge. After incubation, the luminescence measurement in RLU is obtained for each well. In order to be able to compare the 36 plates, it is necessary to reduce the noise due to the experimental bias, and for that, the data are normalized. I have developed a method of normalization called the C-score. This is an analog of the inhibition calculations, but corrects the measurement of each well by the control present on the same lane rather than by the average of the control of the plate. The selection of the hits is done by a mix of threshold methods and Bayesian methods on the BHTS pack. The Bayesian methods take the normalization data as input and output the probability for each compound to be a hit. A threshold matrix implemented in this package allows us to say that if the probability of a compound is greater than this threshold, the compound is a hit. To identify compounds that could be used in therapy, we first compare hits that kill cells from AML KMT2A AF9 patients and not CD34 positive control cells, identified by BHTS pack and C-score. For the identification of a hit by BHTS pack, the conditions are as follows. Be a hit for at least one of the patients and not be a hit for CD34 positive cells. For the identification of hit by C-score, the conditions are as follows. Have a C-score for CD34 positive cells less than 30% and for at least one patient greater than 70%. To be more stringent, the compound should show a difference in C-score between CD34 positive cells and at least 2 patients greater than 50%. We then validated the compound identified by the 2 methods by a dose response screen. The hit mark show the dose-response screen for 60 compounds from the FDA-approved library testing on a KMT2A-AF9 cell lines in 10 dose in duplicates, colored by the inhibition values. Among these compounds, we have identified a compound X involved in nucleotide synthesis, which shows specific activity for AML KMT2A-AF9 fusion not yet studied. The figure below shows the dose-response curve of compound X for 2 KMT2A-AF9 cell lines and the CT34 positive cells. To conclude, this work will help advance our understanding of the biology of acute myeloid leukemia, but will potentially help identify new compounds that could be used for therapeutic purpose in patients, as well as in the development of clinical diagnosis. Thanks for your attention.